ওইটুকু একটু বানাতে দেবো। বাবা বাবা ping ping, देखो ping ping, आधा आई है ना, ये भैया, ये ना ना, ये मार खाली मार खा खा डाउन खाओ লাইভে আছে বুঝছো ওকে জাস্ট 1 মিনিট আমি স্ক্রিন শেয়ার করছি ল্যাপটপে চার্জার
Okay, hello everyone. আমি স্ক্রিন শেয়ার করছি, দেন আমরা ডিসকাশন করব। আজকে বায়োকেমিস্ট্রির কিছু ইম্পর্টেন্ট পার্ট কার্বোহাইড্রেট মেটাবলিজমের আমরা সেগুলো নিয়ে আলোচনা করব। আজকে আমরা অলরেডি কার্বোহাইড্রেট মেটাবলিজমের বেশ কিছু পড়েছি। আমি আজকে এখন ক্লাসটা যেহেতু ইউটিউব লাইভ হচ্ছে, ঠিক আছে, তো এটা আমি বাংলা, হিন্দি, ইংলিশ তিনটা মিলিয়ে বলবো। মেনলি আই ট্রাই টু ডিসকাস ইন হিন্দি অ্যান্ড ইংলিশ মেনলি। ওকে, আচ্ছা। সো মাই রিকোয়েস্ট টু এভরিওয়ান যে আমি গ্রুপের মধ্যে অলরেডি দিয়েছি। সো ইফ পসিবল ইউ ক্যান শেয়ার দ্য লিঙ্ক টু ইউর ফ্রেন্ডস। ওকে, যাতে আরও অনেকের কাছে পৌঁছে যায়। ঠিক আছে, তো অসুবিধা নেই। তো আমরা স্টার্ট করছি আজকের ক্লাসটা। ওকে, আই হোপ প্রত্যেকের কাছে স্ক্রিনটা ভিজিবল। এখানেও স্ক্রিন শেয়ার করে দিচ্ছি ইউটিউবে। ওকে, এক মিনিট। Okay, so first of all, yeah, so it is visible. Okay, so basically previously we have discussed about the carbohydrate metabolism of different pathways in carbohydrate metabolism, as also as well as like different pathways like the glycolysis, like TCA cycle, like ED pathway, infinite pathway, like pentose phosphate pathway. So we have discussed it previously. So today also I just give you some summary about the whole carbohydrate metabolism, and also we'll discuss some other pathways. Okay, so that are very important, and we will summarize totally the total carbohydrate metabolism today. Okay, so first of all, just 1 minute. Yeah. First of all, I just want carbohydrate metabolism. That then what is metabolism and how can we classify it, as well as we will discuss about the different type of catabolic pathways, different anabolic pathways, and so on. Okay, okay. So I hope you all can see the picture. You all can see the uh, handwritten notes here. So basically, in my coaching institute, I always try to give some handwritten note like this. Okay, and also I basically provide some PYQs and also discuss the whole syllabus as much as possible. So first of all, just a brief idea I would like to give, that what is metabolism and what are the classification of metabolism. So metabolism, you all know that the the formation of the molecule of the biomolecules inside of the each and every cell of our body. Okay, as well as the breakdown of the biomolecules to different products. So the formation and the breakdown, okay, both are, if we togetherly called it as the metabolism. Okay, so the breakdown of the biomolecule is called the catabolism. Okay, inside each and every cell of our body system. যে একবার একটু দেখো না যে ইউটিউবটা চালিয়ে যে আমি সাউন্ড আসছে না প্রপারলি, আমাকে অফ রাখতে হয়েছে। And also in the anabolism, the formation of the product, the formation of different type of products, different type of biomolecules inside of each and every cell is called the anabolic pathway, anabolism. So there are some catabolic pathways, there are some anabolic pathways, and togetherly we call it as the metabolism. And there are some intermediate steps, okay, like the TCA cycle, tricarboxylic acid cycle, or the glyoxylate pathway, or the formation of acetyl CoA to pyruvic acid, the pyruvate, and the formation of oxaloacetate from pyruvate. So these all are some the intermediate steps which basically uh, relate the catabolic pathways and the anabolic pathways somehow. Okay, so when we'll discuss about the TCA cycle, you will be able to see that some catabolic pathways are directly linked with some anabolic pathways also. Okay, or from some catabolic pathways, different other metabolic pathways can start. Like from the carbohydrate metabolism, different amino acid metabolic pathways can start. Some fatty acid metabolism can start. So all the carbohydrate metabolism or amino acid metabolism or lipid metabolism, so all are interlinked with each other. So it's a community, it's a networking, okay, between the metabolic pathways. Okay, so today we'll discuss about the classification of the metabolism. So here you can see that the catabolism, anabolism, and the intermediate steps. And again, the classification of the catabolism, you can see we can classify the catabolism, the catabolic pathways into glycolysis, okay, as well as the glycogenolysis. So lysis means the breakdown, okay, lysis means breakdown. So glycolysis, glucogenolysis means glycogen breakdown is called the glucogenolysis. But glycogenolysis, not glucogenolysis exactly, it is called glycogenolysis, mean breakdown of glycogen to glucose. And glycolysis means the breakdown of glucose to pyruvic acid. Okay, then fructose metabolic pathway, so breakdown of the fructose. So galactose metabolic pathway, breakdown of the galactose. Okay, ED pathway, internalization pathway, I've already discussed it, which is also known as the KDPG pathway or 2 keto 3 deoxy 6 phosphogluconate pathway. Okay, so these are the catabolic pathways in carbohydrate metabolism. So if we talk about the anabolic pathways, so there are some formative pathways. We can also call it as the formative pathways. Like that is why it is called the anabolic pathway or the anabolism. Like the glycogenesis. So here you can see the term genesis. Okay, genesis means the formation. Okay, genesis means formation. So glycogenesis means the formation of glycogen from glucose. Gluconeogenesis, synthesis of glucose from different amino acid of the non-carbon precursors. We'll see it today. There is the gluconeogenesis or neoglucogenesis, and the pentose phosphate pathway. So formation of the pentose sugars, like the ribose sugar. Okay, uh, so the formation of the pentose sugar, that is why it is called the pentose phosphate pathway. Okay, so very important. this catabolic pathways. So I hope you can understand there are different catabolic pathways which are listed here, as well as there are some anabolic pathways which are listed here, and also there are some intermediate steps, and the most important one is the tricarboxylic acid cycle or the TCA cycle. So in the TCA cycle, we call it the TCA cycle. Basically, is a very important pathway because from where the maximum amount of energy in the carbohydrate metabolism are produced. That is the ATP. The formation of ATP is produced in a high amount in the tricarboxylic acid cycle or in the carbohydrate metabolism. Okay, like there are basically 24 ATP are produced in the TCA cycle. So when we talk about the glycolysis, so glucose gets converted to pyruvic acid, and for the production of pyruvic acid, two molecule of ATP is being produced. Okay, if we don't consider the oxidative phosphorylation, then we can say that two molecule of ATP are produced. But if we consider the oxidative phosphorylation, okay, the phosphorylation step, then the number of ATP produced in glycolysis is 8 in number. Okay, 8 in number. So in glycolysis, the formation of ATP is 8, and in TCA cycle, the formation of ATP is 24. If we consider the oxidative phosphorylation step, so that means in whole total it is 24 plus 8 equals to 32. Always remind that I'm talking about when we are considering the oxidative phosphorylation step. Okay, in glycolysis as well as in the TCA cycle, then only the amount of ATP will be counted as 28 plus 24 equals to 32. So some of you can ask me that, sir, in previous classes, in school level also, we learned that the number of ATP produced is 38 in number. Then how it is 32? How it is 8 plus 24 equals to 30? Pyruvic acid is converted to acetyl CoA in the aerobic fate of glycolysis. I have already discussed in the previous class. So when the pyruvic acid gets converted to acetyl CoA with the help of an enzyme complex called the PDH complex, pyruvate dehydrogenase, which is basically made up of three enzymes and five coenzymes, sir. Okay, the enzyme one is called the pyruvate dehydrogenase, enzyme two is the dihydrolipoyl transacetylase, enzyme three is dihydrolipoyl dehydrogenase. Okay, so these are the enzymes, and the coenzymes are the TPP, thiamine pyrophosphate, CoASH, FAD plus, NAD plus, and lipomide. So these are the coenzymes, and the enzymes are, I have already discussed, that is the U1, E2, E3. So when this all together, these three enzymes and five coenzymes works together, it makes the PDH complex or pyruvate dehydrogenase complex, which is responsible for converting pyruvic acid to acetyl CoA. And that acetyl CoA is responsible for the beginning or for the starting of the TCA cycle. So when we have discussed about the TCA cycle, you can see that the initial steps are being carried out by two reactant called the acetyl CoA, and the one is oxaloacetate. So when the oxaloacetate and the acetyl CoA comes together and reacts with each other, it makes the citric acid or citrate. Okay, so I have already discussed this in the previous class in the TCA cycle section. So I just want to say that that intermediate part, which is responsible for the production of acetyl CoA from the pyruvic acid, that I have already mentioned here in the third point, that is the formation of acetyl CoA from pyruvate. Here, the remaining 6 ATP is produced, and that is why total amount of ATP produced during the glycolysis and up to from glycolysis to TCA, including the intermediate step, it is 8 from glycolysis, 6 from the intermediate step, and remaining 20 from the TCA cycle. So whole total it is 8 plus 6 plus 24, that is 38. So in that respect, 38 molecule of ATP are being produced when we consider from glycolysis to TCA cycle. Okay, so that is why these intermediate steps are very important. And another important thing is that the pathways like TCA cycle, which can correlate or which can yes, correlate to different pathways. Okay, that means if we see the TCA cycle, then we can see that there are some product produced which is responsible for beginning of any other pathway in different metabolic pathway. Like so a carbohydrate, the TCA cycle is a carbohydrate metabolism, is a part of carbohydrate metabolism. But when the product of TCA cycle start or initiate the any other metabolic pathway, like amino acid metabolic pathway, like lipid metabolic pathway, like the fatty acid metabolism. Okay, so this kind of linking step, so that is why this TCA cycle is called the linking step. Why? Because it can link to different pathway, it can link to different pathway, and that is why this kind of pathway is called the anapleuritic reaction. That is why you can call it as the anapleuritic reaction. As well as this is the pathway, this is the TCA cycle, is the pathway where both the formation as well as the breakdown both occur simultaneously. So formation of some product and the breakdown of some product both are occurring simultaneously. So that is why we can call it also the amphipathic pathway. Okay, that is why both the formation and the breakdown both are occurring simultaneously. So that is why this TCA cycle is so much important, because it can interlink to different pathways, as well as it is the pathway where both the catabolism as well as anabolism, that is the formation of some product as well as the breakdown of some product, take place simultaneously. So that is why this TCA cycle is so much important. Okay, in respect of ATP formation, in respect of connecting different pathways, in respect of formation and breakdown simultaneously of different products. So this is one of the most important pathway in carbohydrate metabolism. And also I have discussed the intermediate steps where pyruvic acid gets converted to acetyl CoA with the help of enzyme complex PDH complex. And I have discussed that six ATP, six molecule of ATP are being produced in the TCS in the intermediate steps. And that is why if we consider the oxidative phosphorylation, then the glycolysis, the total formation of ATP will be 8. In intermediate step, the total formation of ATP will be 6. And in the TCA cycle, the formation of ATP will be 24. So in whole total, the formation of ATP will be 38 in number. So from point of view, there are so many questions can arise. There are so many MCQ can come from this section that I have already discussed. That which one of the following pathway is known as the anapleuritic pathway? Which one of the following pathway is responsible for the formation is involved with the enzyme complex? Which one? So these are the question may come in the exam. How many ATP is produced during the carbohydrate metabolism when we consider the oxidative phosphorylation step? So these are the questions that may come during your examination. Any examination it can come. Okay, I always tell you that if you consider the oxidative phosphorylation, I'm stressing it because the oxidative phosphorylation is a very important step. The oxidative phosphorylation is very important step. So that is why if you don't you should learn about the oxidative phosphorylation very well. Otherwise माने गलत हो जाएगा आंसर. ठीक है, तो इसीलिए ऑक्सीडेटिव फॉस्फोराइलेशन को अच्छे से जानना पड़ेगा पहले. Okay, so now I have already discussed about the glycolysis, so I'll not discuss about it today. I have already discussed about the TCA cycle also. I have discussed about the Entner-Dodrop pathway. I have discussed about the pentose phosphate pathway. So I have discussed about these pathways already. So that is why I will not discuss about it. Okay. So the pathway that I will discuss. Here is the anaerobic respiration of glycolysis. I have already discussed about it. That anaerobic respiration means the fate of glycolysis in anaerobic condition. That means when the pyruvic acid are produced, then what will in which product the pyruvic acid will be converted in the eukaryotic as well as the prokaryotic system. So if in the eukaryotic system the pyruvic acid will be converted to lactic acid, in absence of the oxygen. Okay, so here you can see the during the glycolysis, the glucose gets converted to pyruvic acid or the pyruvate, and the pyruvate is converted to the lactic acid. The pyruvic acid is converted to lactic acid with the help of a single enzyme called the lactate dehydrogenase. With the help of a single enzyme called lactate dehydrogenase. Okay, so 1 minute, just 1 minute. Mm, okay, just 1 minute. Give me 1 minute. Just 1 minute. Okay. Mm, 1 minute, 1 minute, 1 minute. Okay, okay, yeah. So the for the conversion of the anaerobic condition, the anaerobic fate of glycolysis, you can see that the glucose is converted to pyruvic acid, and the pyruvic acid will be converted to lactic acid with the help of a single enzyme called the LDH or lactate dehydrogenase. So this enzyme is a very important enzyme, and why? Because this enzyme is a special character. Do status. Okay. So this LDH is also known as the isoenzyme or isozyme. Okay, why? So what is isozyme? So from this part, the question may come that what is isozyme? The enzymes which are structurally different but functionally same. The enzymes which are structurally different but functionally same, okay, is called the isoenzyme or isozyme. So LDH is one of them. So there are five different types of LDH is present. LDH 1, 2, 3, 4, 5. So these all five enzymes are basically isozymes of each other. Okay, so that is why this enzyme is very important. So the question may come from this also. Okay, the LDH1, LDH2, LDH3, LDH4, LDH5. So these all are the enzymes that are structurally different but functionally same. So that is why its importance is it is very important enzyme. Okay, next. So this is the fate in the eukaryotic system I have already discussed. Okay, the another is in prokaryotic system, the fermentation. In prokaryotic system, fermentation takes place and the alcohol is produced in the prokaryotic system from the pyruvic acid, as well as lactic acid can also produce in the prokaryotic system. Okay, for example, here you can see that in prokaryotic system, glucose first convert to pyruvic acid, that is pyruvate, and from pyruvate to alcohol, and it is a two-step process. Okay, so if we start from the pyruvate, you can see the pyruvate decarboxylase enzyme in presence of TPP, thiamine pyrophosphate coenzyme, okay, convert the pyruvate to acetaldehyde and carbon dioxide, because decarboxylation take place. So you can see that the pyruvic acid is a three carbon molecule. Okay, so when the decarboxylation take place, the C minus, that is CO2, is released. So one carbon is released, so that is why remaining two will form the acetaldehyde, and that acetaldehyde will be converted to ethanol with the help of enzyme alcohol dehydrogenase. Okay, so NADH plus H plus will come and provide some hydrogen. Okay, and that will convert to ethanol or CH3CH2OH. So very important. So alcohol is produced in the prokaryotic system, which is also, we call it as the fermentation process. Okay, and in some of the bacteria, along with this ethanol production, some other product can also be produced, like lactic acid. Okay, in prokaryotic system also, they can also produce the lactic acid. So depending on that, we can divide the prokaryotic system in two sections. One is called the homolactic fermenter, and another one is called the heterolactic fermenter. So homolactic fermenters are the microbial system that are capable of producing only a single product, that is the lactic acid. Okay, that is the lactic acid, because that is why it is called homolactic fermenter. So that they will uh, produce the lactic acid only. For example, E. coli, for example, Zymomonas mobilis or mobili. So these are the homolactic fermenter, can produce only the lactic acid. Okay, again, if we talk about the heterolactic fermenter, for example, the Acetobacter aceti is an example of heterolactic fermenter. It can produce lactic acid, as well as it can also produce the ethanol. Okay, it can also produce ethanol. So lactic acid and ethanol both can be produced by this microbial system, and that is why it is called as the heterolactic fermenter. So more than one product is being produced by this microbial system. So that is why it is called heterolactic fermenter or heterolactic fermentation, because both the system produce more than one product. Okay. Next, if we talk about the aerobic respiration of glycolysis, or the aerobic fate of the pyruvate or the glycolysis, then you can see I have already discussed it, that the enzyme complex is here, that is called the PDH complex, or the pyruvate dehydrogenase complex, which is a multi-enzyme complex, and is composed of three enzymes and five coenzymes. That is the pyruvate dehydrogenase, dihydrolipoyl transacetylase, dihydrolipoyl dehydrogenase. There is the E1, E2, E3. There are three enzymes. These are the three enzymes, and the five coenzymes are the TPP, CoASH, lipomide, FAD+, and NAD+. So these three enzymes and five coenzymes togetherly called the enzyme complex, or the PDH complex, which is responsible for converting pyruvic acid to acetyl CoA, which is the starting reactant of TCA cycle. Okay. So here in the reaction, you can see the conversion of pyruvate to acetyl CoA. So pyruvic acid converts to the acetyl CoA in 4 to 5 steps. In first step, the pyruvic acid gets decarboxylated by the decarboxylation reaction, and in presence of the TPP, thiamine pyrophosphate, and with the help of enzyme E1. Here also, the decarboxylation, that means the carbon dioxide will be released from the pyruvic acid, and the TPP will bind with it to make the acetyl TPP. Okay, then this acetyl TPP, in presence of enzyme 2, that is the dihydrolipoyl transacetylase, and in presence of lipomide and SH-CoA, the TPP will be released from the reaction, and the acetyl CoA will be produced. But along with the acetyl CoA, reduced lipomide will produce. So here you can see the lip bond SH bond SH, that is the reduced lipomide. And then again this reduced lipomide will be converted in a to will be converted to normal lipomide, okay, in presence of enzyme 3 or the dihydrolipoyl dehydrogenase. And here the FAD plus comes and take up that 2 hydrogen from the lipomide, and the lip bondage will be produced, and the FADH2 will be produced, and that FADH2 will that from that FADH2 2 hydrogen will be taken up by the NAD plus, and that FADH2 will convert to FAD plus and will recycle to the reaction. It will back to the reaction. But the NADH plus H plus that is produced during this pathway, during this intermediate reaction, this will go to the electron transport chain and will produce 6 molecule of ATP. I have already discussed that the in this intermediate step 6 molecule of ATP is produced. Why I am calling it the 6 molecule of ATP? Because the pyruvic acid here is the 2 number of pyruvic acid is there, because the end product of glycolysis is 2 number of pyruvic acid. So 2 pyruvic acid are produced in the last step of glycolysis. Okay, so whatever will occur after that will be in 2 in number. So here also the 2 pyruvic acid will be converted to 2 acetyl TPP, 2 acetyl TPP will be converted to 2 acetyl CoA, 2 acetyl CoA will be converted to along with this 2 acetyl CoA, whatever is produced all in 2 in number. So that is why 2 NADH plus H plus will be produced, and that we all know that for one every single NAD plus NADH plus H plus 3 molecule of ATP generally produced in the electron transport chain. So if there are 2 NADH plus H plus is there, then 3 into 2 equals to 6 amount of ATP will be produced in the electron transport chain, and that is why the amount will be 8 in glycolysis, 6 in this intermediate step, and 24 in the TCA cycle along. So all in total 38 in number, 38 molecule of ATP will be there. Okay, so this is the intermediate step, very important. And another one is the conversion of pyruvate to oxaloacetate. So I just want to clear one thing here. So the pyruvic acid which is to oxaloacetate, that I have already written here, is not the pyruvate that produced in the glycolysis. Okay, this pyruvate is from different other pathways like the amino acid metabolism or the fatty metabolism. From that, the pyruvate produced is converted to acetate, but not from the carbohydrate metabolism. The pyruvic acid which is produced during the glycolysis or any kind of other pathways in the carbohydrate metabolism, or mainly the glycolysis, I'm talking about the glycolysis pyruvic acid, is not converted to oxaloacetate, because the pyruvic acid which is produced in the glycolysis in the end product as an end product is generally converted to acetyl CoA, not the oxaloacetate. The pyruvic acid which is converted to oxaloacetate are basically the product of some other pathways. Okay, so that is why, so you should clear that concept. And the very important enzyme is the pyruvate carboxylate, pyruvate carboxylase. That means carbon dioxide will bind with it, pyruvic acid, to make the oxaloacetate. Oxaloacetate is a 4 carbon product, okay, and pyruvic acid is a 3 carbon product. So this 3 to 4 carbon conversion can by this enzyme pyruvate carboxylase. Okay, so I have also discussed about the TCA cycle previously, so I'll not discuss about it today. Okay, just 1 minute. So these are the enzymes which acts in the TCA cycle. So very important enzymes are there. So we also learn about these enzymes: citrate synthase, aconitase, isocitrate dehydrogenase, alpha ketoglutarate dehydrogenase, succinyl CoA synthase, succinate dehydrogenase, fumarase, malate dehydrogenase. So these are the enzymes which is responsible for the TCA cycle. Okay, so I have already discussed about these things. So all the question answer is produced in the is provided in this handwritten note that how many ATP are there, how many ATP are produced here. Okay, so today what we will study, so this is the internal tutor of pathway, ED pathway. So all the pathways that are provided in the handwritten note. This is the pentose phosphate pathway or hexose monophosphate shunt. So these enzymes in the pentose phosphate pathway, transketolase and transaldolase, in every quick exam, in every examination, so this transketolase, transaldolase, this 100% will come. That how many carbon atoms are transferred by the following enzymes? Okay, transketolase or transaldolase, and in which reaction the transaldolase or the ketolase take part. Okay, so very important. So these are the two questions that may come in the different examinations. Okay, so you should learn about it very well. Transketolase, transaldolase, transketolase. So transketolase is a two carbon transporter, टू कार्बन ट्रांसपोर्ट, ये दो कार्बन ट्रांसपोर्ट करता है. देखिए C5 से C3 C7 बना रहा है, मैंने दो कार्बन यहाँ से उठ के यहाँ पर बैठ गया, तभी जाके C3 एंड C7 बन गया. ओके, एंड देन ट्रांसएल्डोलेज C3 C7 को ऐड करके बन रहा है C6 C4. दैट मीन्स यहाँ पर C3 C7, C3 इज कन्वर्टेड टू C6 एंड C7 इज कन्वर्टेड टू C4, माने तीन कार्बन ट्रांसपोर्ट हो रहा है. Okay, so transaldolase is responsible for three carbon transporter. Okay, तीन कार्बन ट्रांसपोर्ट होता है ट्रांसएल्डोलेस के माध्यम से, एंड ट्रांसकीटोलेस के माध्यम से दो कार्बन ट्रांसपोर्ट होता है. ओके, सो वेरी इंपॉर्टेंट, एंड इट हैज टू path phases, oxidative phase and the non-oxidative phase. So the important pathway is the electron transport chain, very important, VBVBI. ओके, यहाँ से बहुत सारे क्वेश्चन आता है. ये इलेक्ट्रॉन ट्रांसपोर्ट चेन, ये जो मैं यहाँ पर ड्रॉ किया हूँ, इसको अगर आप लोग अच्छे से देखेंगे, ठीक है, तो मैंने बहुत कुछ एक साथ हो जाएगा. ठीक है, क्योंकि ये इलेक्ट्रॉन ट्रांसपोर्ट चेन का सबसे बढ़िया पिक्चर है. ठीक है, जहाँ पर सब कुछ डिफाइन किया गया है, सब कुछ A to Z, कि कौन से मैंने इलेक्ट्रॉन ट्रांसपोर्ट चेन क्यों होता है, क्यों इलेक्ट्रॉन ट्रांसपोर्ट चेन में कितना, मैंने कैसे इलेक्ट्रॉन ट्रांसपोर्ट होता है, कैसे वाटर मॉलिक्यूल जनरेट होता है, कैसे एटीपी जनरेट होता है, कौन से सब यूनिट का कौन सा नाम है, दैट इज प्रोटीन यूनिट का कौन सा नाम है. यहाँ से बहुत सारा क्वेश्चन आता है. ठीक है, इसको हम कैसे इनिबिट करें. ठीक है, एक मिनट. तो यहाँ से बहुत सारे क्वेश्चन आता है. ठीक है, तो यहाँ से बहुत सारा क्वेश्चन आता है, जो मैंने क्वेश्चन मैंने 100% हर साल इलेक्ट्रॉन ट्रांसपोर्ट चेन से आप एमएससी एंट्रेंस दो, कहीं पर भी दो, या फिर जो गेट है, गैट भी है, सीवीटी है, और जो नेट गेट सेट जो भी है, ठीक है, आईसीएमआर आईसीआर, सब जगह में इलेक्ट्रॉन ट्रांसपोर्ट चेन से हर साल क्वेश्चन पूछा जाता है. ठीक है, तो पर जो इंपॉर्टेंट मैटर है कि आपको सीक्वेंस बहुत अच्छे तरीके से याद रखना पड़ेगा कि किसके बाद क्या आता है, क्या होता है, कि कौन इलेक्ट्रॉन को कैरी कर रहा है. ठीक है, कौन सा साइटोक्रोम है. ठीक है, कौन से किसका नाम क्या है, इफ इट जो एटीपी प्रोडक्शन जहां पर होता है, जो कॉम्प्लेक्स 5 को हम कहते हैं. ठीक है, एटीपी सिंथेस, क्योंकि वहां पर एटीपी प्रोडक्शन होता है, तो कैसे एटीपी प्रोडक्शन होता है, F0 F1 कॉम्प्लेक्स का नाम क्या है. ठीक है, ये सब कुछ यहाँ से पूछा जाता है यहाँ पर. तो ये पिक्चर बहुत इंपॉर्टेंट है. इसको अगर आप लोग अच्छे तरीके से देखेंगे तो मैंने बहुत सारा क्वेश्चन यहाँ से ही बन जाएगा. ठीक है, तो मैं थोड़ा कोशिश कर रहा हूँ इलेक्ट्रॉन ट्रांसपोर्ट चेन को समझाने के लिए. आई एम जस्ट ट्राइंग माय बेस्ट. सो इलेक्ट्रॉन ट्रांसपोर्ट चेन, इलेक्ट्रॉन ट्रांसपोर्ट चेन होता कहाँ पर है? सो आप यू कैन सी दैट intermembrane space, inner membrane and the mitochondrial matrix are there. So that means it is mitochondria. Okay, so the produce, and that is why you call it as the powerhouse of the cell. Okay, and the ATP produced in the mitochondria, okay, with the help of different cytochromes. Okay, so if you can see the picture, you can see that one, two, three, four, five different protein components are there in the is linked with the inner membrane. And we can call it also the transmembrane protein, because it is present in the inner membrane, the maximum part, and the remaining part present in the matrix or in the intermembrane space. Complex 1, I'm talking about the complex 1. So here you can see the picture. This is the complex 1, then this is the complex 2, this is also the 2, this is the complex 3, com 3, this is the complex 4, this is the complex 5 or F0 F1 complex. So the name of the complex 1, the protein complex 1, is called the NADHQ reductase. NADHQ reductase, which is also known as the complex 1. The complex 2 is known as the succinate Q reductase. Complex 3 is known as the Q cytochrome C reductase. The complex 4 is known as the cytochrome C oxidase. And the complex 5 is known as ATP synthase. So these are the 5 protein components which is a very which is the integral part of the proteins in the electron transport chain in the mitochondria. Okay, so the complex one is basically occupied, is booked for the NADH plus H plus, nicotinamide adenine dinucleotide. So this is basically occupied or the booked by the NADH plus H plus. Why I'm telling like this? Because complex 1 is the place where the NADH plus H plus will come and provide the hydrogens and will release the hydrogen here. So in all the metabolism like glycolysis, like carbohydrate metabolism, amino acid metabolism, lipid metabolism, nucleic acid metabolism, so from everywhere NADH plus H plus are accumulated and comes and release its hydrogen in the complex 1 of the electron transport chain, and it provides its hydrogen to the FMN, flavin mononucleotide, which is then converted to FMNH2, and then again that hydrogen will be provided to FeS or iron sulfur complex. So as this is an electron transport chain, we have to consider the electrons simultaneously with the hydrogen. So if I talk about the 2 hydrogen is provided here, that means 2 electrons are released here. So electron and the hydrogen is nothing but a proton. So the concept of proton and electron will be there. So the number of the number of hydrogen or protons released here, that means the number of electron released here. So we have to have the concept of both the proton and electron simultaneously. So when this FMNH2 release the hydrogen, that means proton, that means electron, to FeS, then FeS then release that electron to Q or ubiquinone. Then Q means ubiquinone, the electron acceptor. It accepts the electron, it carries the electron, okay, and it transfer the electron from complex 1 as well as from complex 2 to complex 3. I have discussed that complex 1 is booked by the NADH plus H plus. In same manner, complex 2 is booked by the FADH2, FAD plus. So in different metabolic reaction in whole the body system, like whether it is a carbohydrate metabolism, it is nucleic acid metabolism, it is lipid metabolism, it is protein metabolism, whatever the metabolism is, some of the FADH2 are accumulated in from all the metabolic pathway, and that accumulated FADH2 will come and release that hydrogens to the complex 2, because the complex 2 of the electron transport chain is booked by the FADH2. So if the FADH2 will come from the different metabolic pathway and will release that hydrogen to the FES, iron sulfur complex. And again, the concept here is the as the proton and electron are, we have to think about the both things simultaneously. That means it is releasing the hydrogen, means it releasing the electrons. Okay, and that electrons will be carried again by the ubiquinone Q, and it will be converted to QH2. So the ubiquinone is the electron carrier, okay, that take up the electrons from the complex 1 as well as from the complex 2. That means it take the proton as well. Okay, electron means the proton here. Okay, so Q is converted to QH2, and that electron will be provided to complex 3. So whatever the electron or the hydrogen is being accumulated or by being transferred in the complex 1 and 2 is collected from the 1 and 2 will be transferred by the ubiquinone to the complex 3. And this complex 3, here you can see the complex 3 is composed of different cytochromes. First, the electrons will be provided to cytochrome B, then that electrons will be transferred to the FES complex. In the complex 3, you can see I'm talking about the complex 3 here. Okay, so the QH2, the electrons, the ubiquinone will provide the electron to the cytochrome B, and then that electron will be transported to the electron FES iron sulfur, and again that electron will be transported to the cytochrome C1. So from the ubiquinone QH2 to cytochrome B in the complex 3, from cytochrome B to FES, from FES to cytochrome C. So complete the this is the complete in uh, complex 3. Then that cytochrome C will provide that electrons to the cytochrome C1 will provide that electron to the cytochrome C, which is the bridge between complex 3 and complex 4, which is the bridge between complex 3 and complex 4. So this cytochrome C will take up that electron and will provide that electron to cytochrome A in complex 4. In cytochrome A, when the cytochrome A receive the electrons, it again transport that electron to cytochrome A3 minus copper 2 plus in the complex 4, and then that cytochrome A3 minus copper 2 plus will release that electron to molecular oxygen in the mitochondrial matrix, and after reacting with the molecular oxygen, the water is produced. So from the complex 4, water is produced. Okay, so this is the process through which water is being produced in our body system, in our cell. Okay, because we all know that our cell, each and every cell is made up of 70% of water. Okay, then so this is the general scenario, but there are some another points, like you can see the complex 1 and complex 3. Complex 1 and complex 3 is a one-way passage for hydrogen. So hydrogen from the mitochondrial matrix can easily transport to the intermembrane space. Okay, but the problem is it is a one-way passage, one-way system. So the mitochondria, from the mitochondrial matrix, hydrogen can cross the inner membrane and can go to the intermembrane space with the help of complex 1 and complex 3, but they cannot come back by the complex 1 and complex 3. So that is a big problem. So as they are not being able to come back from the intermembrane space to mitochondrial matrix, so the concentration of hydrogens are getting increased time by time to time, are getting increased. So at a certain time of point, okay, the number of the concentration of the hydrogen is become so much high that it produce a electrochemical gradient. Okay, proton gradient. It produce a proton gradient, and it produce a force that the hydrogen have to come back to the mitochondrial matrix to equalize the both part. That means as the mitochondrial matrix releases the hydrogen from the mitochondrial matrix to the intermembrane space, that means the H plus ion concentration is getting lower in the mitochondrial matrix section, and the H plus ion concentration is getting higher in the intermembrane space. So there is a disbalance between these two sections in the mitochondria. So that is why at a certain point of time, a proton motive force will be generated. A proton motive force will be generated by the H plus ion. That means the hydrogen have to come back from the intermembrane space to the mitochondrial matrix. For that, for that, complex 5 plays an important role to return back the hydrogen from intermembrane space to the mitochondrial matrix. For that, the ATP synthesis is there. ATP synthesis is basically made up of F0F1, is also known as the F0F1 complex, and is made up of Fernandez membrane. Okay, it is also known as the Fernandez membrane, F0F1 complex, and which is responsible for the production of ATP also. So here you can see the F1 part of the F0F1 complex contains some segmented area like alpha subunit, beta subunit, gamma subunit. The middle part of the F1 is the gamma subunit, and the remaining part, the side by side part, is alpha beta, alpha beta, alpha beta. So the alpha section and beta section is surrounding the gamma section. Okay, gamma is present in the middle, and it is a hollow tube like structure, and surrounding the gamma section, the alpha and beta, alpha and beta, alpha and beta is there in the F1 complex. So it will just like a, so it will look like this one. Okay, so and the alpha and beta part contains pi, inorganic phosphate, and adenosine diphosphate, ADP. So when the huge amount of pressure is produced, এক মিনিট একটু, হ্যাঁ ওরা এখানে গেছে, বাথরুমে গেছে, তুমি ক্লাস নিচ্ছি আসলে, হ্যাঁ পরে কথা বলছি। হুম। Okay, so as the concentration is higher in the intermembrane space, so there is a force produced there called the proton motive force, and that proton motive force will pressurize the ATPase or the F0F1 complex in such a way that the hydrogen will released or will come back from the intermembrane space to mitochondrial matrix by this F0F1 complex. Okay, and when the hydrogen comes back from the F0F1 complex to the mitochondrial matrix, this F1 complex, this F1 is basically start to rotate. When the hydrogens are coming back from the intermembrane space to the mitochondrial matrix, the F1 part of the F0F1 complex start to rotate. And as it start to rotate, the pi, inorganic phosphate, and the ADP, adenosine diphosphate, which is present in the alpha and beta part, okay, of the F1 complex, will bind together to form the ATP. Okay, will bind together to form the ATP. So this is the mechanism through which ATP is produced by the ATP synthase in the electron transport chain. So we all have to memorize the name of the protein complex here. The protein subunits like the NADH-Q reductase complex 1, the complex 2 is the succinate Q reductase, complex 3 is the Q cytochrome C reductase, complex 4 is the cytochrome C oxidase, and complex 5 is the ATP synthase. F mean Fernandez membrane, and F1, from F0 F1 ATP are produced, and from complex 4 water molecule is produced. And when the hydrogen comes back from the intermembrane space to the mitochondrial matrix, then the balancing is there. So in the lab, when we do some research in the mitochondrial research, the laboratory having the mitochondrial research, they basically provide some chemicals which can carry that hydrogens, okay, and can come back and can take up the hydrogen and can cross the intermembrane space to the mitochondrial matrix by by any part, okay, any part of the electron transport chain, and they are called, for example, 2,4-DNP, dinitrophenol, 2,4, which is also known as the uncouplers, okay, which is called the uncouplers. 2,4-dinitrophenol is an example of uncouplers that can go to the intermembrane space, can take up the hydrogen, and can easily come back from the intermembrane space to mitochondrial matrix to reduce the proton motive force there, to reduce the proton gradient there. Okay, so this is very important. This is very important. So we have to know about the uncouplers. So these are the chemical molecules that can cross, that can easily cross the mitochondrial inner membrane layer, but and can go to the intermembrane space and take up the hydrogen, and can easily come back from the intermembrane space to the mitochondrial matrix, and can reduce the pressure, proton pressure there. Okay, so that is why they are called the uncouplers. And coming back to the when they come back to the mitochondrial matrix, they release that hydrogen. Okay, that is why they are called the uncouplers. So coupling, uncoupling is there. So coupling, uncoupling is there. So here you can see the 2,4-DNP when binds phenol and comes back and cross the inner membrane and comes back to the mitochondrial matrix and release the hydrogen there in the mitochondrial matrix. That is why they are known as, that is why they are called the, sorry, they are, that is why they are called the uncouplers. Okay, another one important question comes from this part, that what is PO ratio? PO ratio means phosphate oxygen ratio, that how many phosphate, how many high energy phosphate like ATP is synthesized per atom of oxygen is called the PO ratio. How many? Because we can see that in complex 5 ATP is produced. So how many ATP are produced per utilization of oxygen in complex 4? Because complex 4 is the place where oxygen is used to make the water molecule. So how many oxygens are used to make the, how many number of ATP? This ratio is called the PO ratio or phosphate oxygen ratio. Very important question. Another important question from here is the inhibitors of the oxidative metabolism or the electron transport chain. Very important question. 100% you will get question from this part. 100%. That is, they are called the electron transport blocker. They are called the electron transport blocker. So complex 1 is blocked by rotenone. So if rotenone blocks the complex 1, ma phone করেছিল একটু দেখো তো, if the rotenone blocks the complex 1, okay, if the complex 1 is blocked, think that complex 2 is open, complex 3 is open, then electron will be transported, because complex 2 is there to transport the electron through the ubiquinone. So the water molecule will be produced, but the proton gradient will be less because complex 1 is blocked by the rotenone. So less ATP will be produced if complex 1 is blocked by the rotenone. Now complex 2 is blocked by the malonate, and that malonate, if complex 2 is blocked, complex 1 is open. So complex 1 is open, so electron transport is there. So as electron transport is there, so moderately high amount of ATP proton will be there, and the ATP synthesis obviously will be there. No problem in ATP synthesis. But when the complex 3 is blocked, when the complex 3 is blocked by the antimycin A, then it will not be able to produce the water molecule, because complex 3 अगर ब्लॉक हो गया तो फिर complex 4 में तो जा ही नहीं पाएगा इलेक्ट्रॉन. तो अगर कॉम्प्लेक्स 3 और 4 में इलेक्ट्रॉन नहीं जा सकता है तो फिर वाटर कैसे बनेगा? वाटर तो बनता है कहां से? कॉम्प्लेक्स 4 से. सो नहीं बनेगा. But the proton gradient will be there because complex 1 and 1 is there. So we know that complex 1 से mitochondrial matrix से intermembrane space तक हाइड्रोजन जा सकता है, तो प्रोटॉन तो बनेगा ही. एंड ऑब्वियसली प्रोटॉन बन रहे हैं मतलब प्रोटॉन ग्रेडियंट बनेगा, मतलब कॉम्प्लेक्स 5 में जाके वो फिर से प्रेशराइज करके एटीपी बनाएगा. ओके, सो वेरी इंपॉर्टेंट, कॉम्प्लेक्स 3 भी अगर ब्लॉक हो जाएगा तो वाटर मॉलिक्यूल नहीं बनेगा, पर प्रोटॉन तो बनेगा, प्रोटॉन मोटिव फोर्स बनेगा, फिर हाइड्रोजन, फिर एटीपी भी बनेगा. देन CnCO, साइनाइड एंड CNCO. So this is basically the blocker of complex 4. दिस इज बेसिकली ब्लॉकर ऑफ कॉम्प्लेक्स 4. अगर कॉम्प्लेक्स 4 ब्लॉक हो गया तो फिर वाटर तो बनेगा ही नहीं, क्योंकि वाटर बनता कहां पर है, कॉम्प्लेक्स 4 से. मॉलिक्यूलर ऑक्सीजन के साथ बाइंड करके वाटर बना देता है, पर कॉम्प्लेक्स 4 ही तो ब्लॉक हो गया, माने वाटर नहीं बनेगा. पर कॉम्प्लेक्स 1 इज ओपन, कॉम्प्लेक्स 3 इज ओपन, मींस इलेक्ट्रॉन हाइड्रोजन ट्रांसपोर्ट होगा माइटोकॉन्ड्रियल मैट्रिक्स से इंटरमीडिएट स्पेस तक, वो जाएगा हाइड्रोजन. तो जाएगा तो प्रोटॉन मोटिव फोर्स भी फिर बनेगा. फिर प्रोटॉन मोटिव फोर्स बनने से फिर एटीपी सिंथेस, दैट इज कॉम्प्लेक्स 5 के ऊपर प्रेशर आएगा, फिर एटीपी बनेगा. एंड द लास्ट वन इज कॉम्प्लेक्स 5, दैट इज एटीपी सिंथेस ब्लॉक. एटीपी सिंथेस ब्लॉकर किसको कहते हैं हम? ओलिगोमाइसिन को हम कहते हैं एटीपी सिंथेस ब्लॉकर. नाम पे ही तो बोल रहे हैं कि एटीपी सिंथेसिस को ब्लॉक कर रहे हैं, मतलब कॉम्प्लेक्स 5 को ब्लॉक कर रहा है. तो कॉम्प्लेक्स 5 ब्लॉक होगा पर वाटर मॉलिक्यूल तो बनेगा, क्योंकि वो तो बनता है कॉम्प्लेक्स 4 से. सो कॉम्प्लेक्स 1 भी ओपन है, कॉम्प्लेक्स 3 भी ओपन है, कॉम्प्लेक्स 4 भी ओपन है, कॉम्प्लेक्स 2 भी ओपन है. बट प्रोटॉन ग्रेडियंट बन रहा है. बहुत खतरनाक सिचुएशन है ये वाला, तो बहुत खतरनाक सिचुएशन है. क्यों? क्योंकि प्रोटॉन इंटरमीडिएट स्पेस तो जाएगा पर वहां से वापस नहीं आ पाएगा, क्योंकि कॉम्प्लेक्स 5 ब्लॉक हो गया. तो कॉम्प्लेक्स 5 ब्लॉक हो गया मतलब वहां से वो वापस नहीं आ पाएगा माइटोकॉन्ड्रियल मैट्रिक्स में. तो एक टाइम आएगा जब जब मतलब फट जाएगा, ऐसा काइंड ऑफ सिचुएशन बनेगा. ठीक है. सो दिस इज अ वेरी मैंने हाईली डेंजरस कंडीशन. ओके, अगर हम लोग कोई अनकपलर भेज देंगे तो बच जाएंगे, अगर अनकपलर नहीं भेजेंगे तो सेल फट जाएगा, माइटोकॉन्ड्रिया फट जाएगा. The situation will be like this. Okay, so the oligomycin is very dangerous. So if you use it in the mitochondrial matrix, mitochondrial system, then there is a high chance of danger thing can happen there. Okay, so that is the importance of the inhibitors in the electron transport chain. And the last one is the chemoosmotic hypothesis, which is proposed by the Mitchell. Okay, ये क्वेश्चन भी बहुत आता है. ठीक है, ये केमोऑस्मोटिक हाइपोथेसिस किसको कहते हैं? मिचेल्स केमोऑस्मोटिक हाइपोथेसिस किसको कहता है? प्रोपोज्ड इन 1967. ये थ्योरी इसको कहता है केमोऑस्मोटिक कपलिंग थ्योरी, प्रोपोज्ड इन 1967 बाय द मिचेल्स. ओके, बाय द मिचेल. अब ये कहता क्या है केमोऑस्मोटिक हाइपोथेसिस? कि जो इलेक्ट्रॉन जो आ रहा है ये केमिकल एनर्जी, ओके, माने जो बायोलॉजिकल एनर्जी और जो केमिकल एनर्जी ये जाके एटीपी बन रहा है, केमिकल एनर्जी ऑफ एटीपी में कन्वर्ट हो रहा है. सो द इलेक्ट्रोकेमिकल एनर्जी, इलेक्ट्रोकेमिकल ग्रेडियंट इज कन्वर्टेड टू adenosine triphosphate chemical energy. Okay, so converting the energy which is provided by an electrochemical gradient into the chemical energy of ATP. Okay, so this conversion of electrochemical energy to the this electrochemical gradient to the ATP is called the chemiosmotic hypothesis. Okay, इसको कहता है कि एक एनर्जी से दूसरी एनर्जी में कन्वर्ट हो रहा है, एनर्जी प्रोडक्शन हो रहा है. ओके, so this electroporation system is organized in the redox loop within the membrane, and the electrons are passes from one carrier to another on the respiratory chain. At the same time, the proton are ejected towards the cytoplasmic side with the OH remain on the matrix side. ठीक है. सो ये जो कन्वर्शन हो रहा है एनर्जी में, जो एटीपी प्रोडक्शन हो रहा है, ओके, द प्रोसेस थ्रू व्हिच दिस एटीपी इज प्रोड्यूस्ड, दिस हाइपोथेसिस इज कॉल्ड द केमोऑस्मोटिक हाइपोथेसिस, प्रोवाइडेड बाय मिचेल्स इन 1967 in his chemoosmotic coupling theory. Okay, very important. So this is all about the electron transport chain. So I think मेटाबॉलिज्म में सब कुछ होता है. ठीक है. So in the next class I'll discuss pathways in the carbohydrate metabolism which are very important, and that will be the last class of the carbohydrate metabolism, and I'll discuss about the remaining pathways in the carbohydrate metabolism and their importance in it. Okay, so if you have any doubt then you can discuss it with me. Okay, যদি কোনো ডাউট থাকে তাহলে তোরা জিজ্ঞেস করতে পারিস. আর দিস আর দ্য ভেরি ইম্পর্টেন্ট কোয়েশ্চেন্স. আমি বারবার বলছি এগুলো খুবই ইম্পর্টেন্ট কোয়েশ্চেন, খুবই ইম্পর্টেন্ট জায়গা. এই যে এগুলো যদি প্রপারলি পড়া যায়, যদি প্রপারলি যদি জানা যায়, তাহলে কিন্তু মানে পরীক্ষাতে কোশ্চেন 100% কমন থাকবে মানে এই কনসেপ্টগুলো থাকলেই অ্যান্সার করা যাবে। সো দ্যাট ইজ ওয়াই দিস কোয়েশ্চেন্স আর ভেরি ইম্পর্টেন্ট, দিস কনসেপ্টস আর ভেরি ইম্পর্টেন্ট। এগুলোকে একটু ভালো করে মানে রপ্ত করতে হবে, ব্যাস তাহলেই হবে। ঠিক আছে, তো আজকে এতটাই থাকবে। আবার আমরা নেক্সট ক্লাসে এগুলো নিয়ে ডিসকাস করব। বায়োকেমিস্ট্রি রিমেনিং কার্বোহাইড্রেট মেটাবলিজমের পার্ট নিয়ে নেক্সট সানডে আমি ডিসকাস করব। এবং আমি চেষ্টা করছি যে এখন থেকে যতটা পসিবল হয় লাইভে আসার, এবং এই যে সামনে সব কলেজে অ্যাডমিশন আছে, কোন কলেজে কি মানে অ্যাডমিশন প্রসেস আছে বা কিছু সমস্ত জিনিস নিয়ে আমি খুব তাড়াতাড়ি ডিসকাস করব সবার সাথে। সো যাই হোক, তো আজকের জন্য এতটাই থাকবে। তো যদি কারো কোনো কোয়েশ্চেন থাকে ইউ ক্যান আস্ক মি, আর আদারওয়াইজ আজকে এতটাই আমরা রাখবো। ওকে, সো আমি এখানে ইউটিউব স্ট্রিমিংটা শেষ করছি। থ্যাংক ইউ।